Okay, so today we are going to discuss about the antibodies and its structure as well. So before going towards the structure of the antibodies, we should be known about the antibodies first. Alright, what are antibodies? Let me write here. Okay, so what are antibodies? Antibodies are the protein molecules. So here let me write antibodies are protein molecules. Alright, which are used in the humoral immune response. So let me write that as well. Where are they used? They are used in the humoral immune response. Immune response. Alright, and they are also written as Ig. Wait, let me tell you about the Ig. So here let me write first Ig. Alright, what is this Ig? This Ig means immunoglobulin. Globulin. Alright, which can be abbreviated as Ig, as I've already written. Now, what's the common shape of antibodies? The common shape of antibody, like the common antibody, is Y-shaped. Alright, so let me write here. Y-shaped is the common antibody. Wait, let me write here. This is the Y shape, and this is what? This is the common antibody we do have. This is common antibody. Common antibody. Alright, common shaped antibody. And this is Y shaped. So let me write shaped here as well. Shaped. Alright, so Y shaped antibody is the common antibody we do have. Alright, so now how antibodies are produced? Alright, they are produced by the plasma B cells. Like initially B lymphocytes are differentiated, and these are differentiated into two types: plasma B cells and memory B cells. Memory B cells keep the record like how these antibodies are produced, and how like antibodies are produced by what cells? These are produced by plasma B cells. So let me write here first. So initially when antigen come, wait, let me write here down with another color. Okay. So when antigen come, it triggers our immune system. Alright. It triggers our immune system. Then the differentiation of T lymphocytes, like initially T lymphocytes are activated, then B lymphocytes are activated as well. So I am directly writing here B lymphocytes are activated. So B lymphocytes get activated. Alright, and they are then differentiated into two types of cells. Wait, let me write those as well. Two types of cells. Alright, B lymphocytes are differentiated into two types of cells. What those two types of cells are? Plasma B cells and memory B cells. Memory B cells. Alright, so B lymphocytes are differentiated into two types: plasma B cells and memory B cells. Now what these plasma B cells would be doing? Alright, these plasma B cells are responsible for generation of or production of antibodies. Wait. Responsible for the production of the antibodies. Alright, and what these memory B cells are responsible for? They are responsible for keeping the record, like how these antibodies are made, so that in the future, if that same antigen is entering in your body, so that it should not take time, but instead it should directly make the antibodies. Alright, so these B lymphocytes will keep the record of formation of antibodies. Formation of antibodies. Alright, so if in the future, wait, let me write that as well. In case in future, if the same antigen is a foreign particle, alright, so antigen enters the body, then these memory B cells will be converted into plasma B cells. Alright, so then memory B cells would be converted to the plasma B cells, and plasma B cells are responsible for what? For the production of antibodies. Alright, antibodies. After generation of these antibodies, what will be happening? Like these antibodies will be circulated throughout the body. Alright. Wait, let me write with this color. So ultimately these will be circulated throughout the body. So this was about the antibodies. Alright, so antibodies have a specific part. Alright, they are specific antibodies. They are specific antibodies as well as general antibodies, but here I am talking about specific antibodies. So what are specific antibodies? Wait. Specific antibody or antibodies. Alright, they'll do what? They'll attach to the epitope of the antigen. Attach to the epitope of the specific antigen. Alright, not general but instead specific antigen to kill the pathogen. Wait, let me write that as well. To kill the pathogen. Kill the pathogen. Alright, so why antibodies are used? Antibodies are used to generate immune power. This is the last point. Antibodies are used to generate immune power. To generate Immune power. Immune power. Because we know that when antigen will be entering to the body, it will trigger our immune system, and immune system will be triggered, then B lymphocyte will be differentiated into plasma B cells and memory B cells. Plasma B cells will start producing the antibodies, and memory B cells will be keeping its record, alright, how antibodies are produced. Then these antibodies would be circulated throughout the body. But if in the future that same antigen will be entering to your body, then memory B has already record of production of those antibodies to the specific antigen. Then plasma B cells would be converted into plasma no memory B cells would be converted into plasma B cells then, and plasma B cells again will start producing the antibodies, and then those antibodies will be again circulated throughout the body. So this is how the mechanism of antibodies works. So in the next class we'll be talking about the structure of antibody, which is Y shaped, alright, I have already told you, but we'll be discussing in detail, alright. We'll be discussing about the light chain, long chain, and we'll be discussing about the sulfur loop and sulfur dibond, alright. So we'll be discussing about those all.